Well, good morning, everyone. Welcome to Wednesday, day 2 of class, day 3 of the quarter, and officially the first day of content. Let me just pull up a few things here that I am going to need. Okay. There we go. As always, you want to sign in as you're coming into the class, and we're going to get started. So just a reminder for how we run these Zoom sessions. Today's topic, of course, is the introduction to microbiology that follows chapter 1 in the textbook. Really, the content that you want to be studying is provided in the video lectures and the slides that are posted on the topic page that's labeled Introduction to Microbiology. And this is the page you would have used to prepare for today. So my expectation is that you watch those video lectures, you've reviewed those slides, and you're showing up today with that background. Now, I'm not going to test you on that. And if that ever is not possible, you should still come to class. But if you don't do that prep, you won't get as much out of this class, and I guarantee your grade will not be as high as it could be. All right, so what we're going to do in today's class in Zoom is review some of the key concepts that are provided in this material, but not all of them. So you are responsible for everything in here, even if we don't talk about it in Zoom. And we're going to use our time to try to apply our knowledge to kind of deepen our understanding. So again, I'm assuming you did the basic preparation, and so now you're ready to kind of study it a little bit more, review it, and apply it. So moving forward, let's suppose this week was crazy and you weren't able to do the prep that I asked you to do. You want to make sure that you can work this into your schedule. The topic page is for the stuff that we'll be talking about in the future. And I'll try to remind you at the end of class, often at the end of the week, sometimes at the end of every class, what you should be preparing for next. But if I don't do that, then the schedule is where you go to find out what topics we're approaching. Okay, so let's have a look at this first topic. These are sort of the big questions we'll be looking at. First of all, what is a microbe? Microbiology studies microbes, so what are they? There's 8 official types. We're going to make sure we understand the different features that place a particular microbe in a particular category. Why this field is important. Why do microbes matter? What's their relationship to us? How do we know about them? So a little bit of history. And what are some of the current challenges that the field is facing? So first, our definition from the video lectures, you will have seen that this is an organism or infectious agent that is too small to be seen with the naked eye. So there's lots of things we can't see, but we're going to focus on things that either too small to be seen and are alive, or too small to be seen and cause disease. Sometimes those are the same things, sometimes they're not. An infectious agent being something like a virus, a viroid, or a prion, which are infectious yet not considered to be organisms per se. So that includes things like bacteria, which you might find in your kitchen, and parasites, which are eukaryotic. Here is a Plasmodium parasite in the intestine of a mosquito. This is what causes malaria. Fungi can be macroscopic. If you walk in the forest and you look at mushrooms, you'll see some ones that are not microbes, but there are microbial fungi, and so some members of that kingdom are microscopic. And of course, viruses. If you don't know what a coronavirus is all about, you have been living in a box for the last 2.5 years, and all of us are a little jealous that you haven't had to know too much about that, but I'm pretty sure no one in this class is in that position. There are some fun tools to use, and I encourage you to use them. I'm going to skip through them today, or skip over them today because of time. But it's very helpful to have a sense of scale. So we are talking about things that are real, but not tangible. You can't touch them. You can't see them with your naked eye. So it can be sometimes quite abstract to think about them. I am very comfortable working in the molecular space, the level of DNA and proteins and signaling pathways and how cells talk to each other, because I have been thinking about this stuff for 20 years. But that means I have in my head basically a visual map of all this stuff. When I say a word like transcription, it evokes for me a visual picture of a process that I can see in my mind. So sometimes when you're learning this stuff, it can feel a little bit like it's impossible to remember or understand it. And so it's very useful, I think, to kind of ground yourself in remembering that this stuff is visible. It's just not visible to us. And so going into one of these tools, this is a tool that will zoom you in on the microscopic world, and it kind of helps you to sort of imagine it in your mind. It starts to build that mental map. So when you are feeling lost, go to one of these tools. And if you also just want to amaze your friends and delight your children, you can use these. They're quite cool. I encourage you to do that at the end of class. But here's sort of in words, kind of a sense of how big things are with respect to other things. So a larger microbe would be something that is eukaryotic and single-celled, like an amoeba. So amoebas are typically around 400 micrometers. You can actually see these with your eye, but they look like a speck of dust. Very difficult to know what it is. Under a microscope, much easier to understand it. So this is just under half a millimeter. So still visible to our eyes, but just as a speck. So we still do consider it microscopic. Then we've got eukaryotic cells that are smaller. And this would be some human cells in this range of being like 10 micrometers. It's the smallest human cell. Human cells can be much larger than that. Small eukaryotes include yeast cells, which are members of the kingdom fungi. Below that, at around the 1 micrometer range, so 1,000,000 times smaller than a meter and 1,000 times smaller than a millimeter, are typical bacteria. And then another 10-fold smaller than that is a typical virus. Viruses can be even smaller than this. So because they're invisible, of course, they haven't just been known forever. How long have we known about them? Put your guess into the chat. So this is where you go to your chat and you put in, how long do you think that humans have known about microbes? Maybe you know this because you did the prep. Maybe you did the prep and you don't remember, so you're going to take a guess, which is fine. I just want everyone to participate. If you have to speculate wildly, please speculate wildly, but this will tell me that you're still awake and alive in my class, which are the two most important things. You don't have to know everything. You don't have to have right answers, but being awake and alive is super helpful. And at any time when I verbally ask you a question, you're welcome to pass if it's over microphone, and you're also welcome to put a question mark in the chat if you're just stumped and you don't want to volunteer a guess and you're feeling, unwilling to speculate. That is completely fine, but do participate. Great. I see some question marks too, which is wonderful. Thank you for putting those in. Awesome. Okay. Keep those answers coming. Fabulous. Good job. Okay. So a wide range of answers. So let's start with how long they've known about themselves. I mean, they don't really have self-awareness the way that we do, but they have existed for billions of years. It's been about 400 going on 500 years since we found out that they exist. So in that time, half of that time, we didn't really pay much attention to them. We knew they were around, but then we're like, that's interesting. Moving on. So it's only been in the last 2 centuries that we realized that they are important to us. And now we understand that microbes are crucial to everything that we care about. They're crucial to industry, vital to ecology. They're, of course, an important pathogen that kills people and currently is having a major worldwide impact. They're also very important in the sense that technically you are, in some sense, microscopic because the individual components of your body are cells which cannot be seen with the naked eye. So in summary, life as we know it is because of and is dependent on microbes. And if there's life on other planets, the speculation that it is likely microbial. All right, so one of the things you would have prepared is the history section from the video lectures. And maybe you don't remember everything from that, but we're going to try our best to review it anyway. So again, you're not required to have right answers, but do try to participate in these. All right, so here are your options at top. This is not all of the scientists you have to know about, but this is just a review of some of them. Here are some scientists that you might have remembered hearing about. Which of these do you think was the first to name cells? Put that into the chat. Great. It was indeed Robert Hooke in the 1600s, right? Who of these guys pioneered the use of antiseptics? The use of antiseptics. He was a battlefield surgeon and decided that he would pay attention to what other people are saying about microbes and try cleaning wounds after doing surgery, and voila, people stop dying so much of sepsis. Yes, that's right. And we still have sort of an artifact of him floating around in our medicine cabinet. Listerine, of course, is based on one, it's not exactly the same antiseptic. He used something called phenol, which actually will give you a chemical burn, though that's better than dying of gangrene. But there are phenolics, which are based on phenol, that are used in mouthwash, for instance. Who discovered the very first antibiotic? Who's credited with discovering the very first antibiotic? Right. That's right, that was Alexander Fleming. And what was the antibiotic that he discovered? That's not on this page, but I'm asking you anyway. Yes, that's right. It was famously penicillin. Who is credited with pioneering the technique of vaccination, although some version of this had been around for centuries, actually, and he sort of cribbed it from China. That's right. Indeed. Indeed, it was Edward Jenner in an experiment that is really unethical to perform today. Who is credited with being the first to observe living cells, although one could make a case for other people, but who's usually credited with being the first to observe living cells? That's right. Indeed, it was Van Leeuwenhoek. And who provided strong evidence to support the biogenesis theory of these guys? Lots of people argued back and forth over a couple of hundred years about what was what. He sort of put the nail in the coffin-ish. That's right, it was Louis Pasteur. Not his only claim to fame. And there are important people that are not on this list that you do have to know about. Okay, so more people shown here. All right, so now they're in groups. Okay, this is going to a little bit apply, a little bit of an application question. So let's talk about who did what and when. And so I just want you to look at these names for a minute. So what the groupings are is they're grouped together, not necessarily in terms of time, but in terms of what their discovery has maybe in common. Okay, what their discovery maybe has in common. And so I want you to think about it, look through this and think to yourself, like, what might these guys have in common? Okay, just take a second to think about that. These different groups. So what do these guys have in common? What do these guys have in common? And then we'll do this activity, so hang tight. Okay, so I'm going to cold call somebody. And you're going to give me your best guess as to which of the groups shown here. And I always forget to number them, but you could give them numbers, or you could just say the first person's name. Debated the origins of life, trying to figure out whether it was life arose spontaneously from non-living matter or was generated life from life. So which of these groups? So I'm going to cold call someone, so hang tight. Remember, a reminder that if you are not sure and don't want to speculate, you are always welcome to pass. And if you do want to speculate, that is just fine. Don't be afraid to be wrong. Let's start with Sophia.
I'm gonna guess, because I'm not really sure, but I'm gonna guess maybe the fourth.
1, 2, 3, 4. So Basi, Semmelweis, Pasteur, Lister, and Koch. All right, thank you for your answer. Let's ask class in the chat if you agree or disagree. So just everyone now in the class should participate. Agree or disagree? Excellent. Okay, so we have a variety of answers. Some people agree, some people disagree, and some people are not sure. Okay, so as it happens, that is not the correct answer, but I really appreciate you volunteering a guess, even though you weren't sure. That's very brave, and I always encourage people to do that. Thank you. Please don't be afraid to do that. Let's have a raise your hand for someone who feels confident they have the right answer. Emma, go ahead.
I think it's the second one.
Yes, that is correct. And that was in the 1600s to the 1800s. So over 200 years, people battled this back and forth. All right, next question. Who proposed and tested the presence of endospores? For this one, I am going to cold call Jessica. Again, don't be afraid to speculate. You don't have to be right, but just give us a guess if you can, or you're welcome to pass if you don't want to. Jessica, are you able to unmute?
Sorry.
There you are.
I'm gonna guess Tyndall and Cone.
Yes, that is correct. That was in the 1800s. Good job. Okay. Now I'm going to ask for who investigated the germ theory of disease. Which group of scientists could be grouped into that? And let's go to Laura for this.
Would it be the fourth group?
That's right, in the 1800s. Okay. First to observe cells we've already talked about. So let's have everyone just put that into the chat because I'm expecting you all to know this. Which group of scientists would be the first to observe cells? Yes, good. That's right. The first were the first in the 1600s. And so that leaves our good friends, Jenner, Ehrlich, and Fleming, who developed treatments for disease, pioneered the use of vaccination as it currently exists, developed a drug in the lab based on arsenic called Salvarsan to treat an STD that was killing a lot of people and making lots of other people uncomfortable and miserable. And Fleming, of course, antibiotic penicillin discovery within about 150-year span. Okay, so now we're gonna have a raise your hand. I'm gonna give you a chance to think about this one. From the prep, you would know the answer to this. You're repeating Pasteur's swan-necked experiment, but you keep getting a result that is different than his. So his result sort of helped to establish for most people that biogenesis is how microbes come to be, as opposed to spontaneous generation. But when he published his work and lots of people were convinced, there were people who tried to replicate his data and some of them were not successful. Okay, so if you remember this, what was his result that allowed a lot of people to believe what, biogenesis is the way things go? And why would some people have not had the same result? Raise your hand if you can sort of talk about this. Heather, go ahead.
So his result was he had the S-shaped flask and he wasn't getting any growth in it, but when he broke it, there was growth. And then the people who tried to replicate it, they had like heat resistant microbes.
That's right.
And so, yeah.
Yeah, so he was doing this experiment where he would take some broth and boil it and show that when it was boiled, nothing grew as long as new microbes were not being introduced from the air. But when some people tried to do this work and boiled their broth, things would grow even though there was not microbes coming in. So that was a bit of an issue until Tyndall and Cone kind of figured out what was going on, right? Tyndall proposed that it was because the people whose experiments were failing were using different sources of microbes. He figured out their broth wasn't a yeast broth, it was a hay infusion, which has a lot of soil microbes, and soil microbes are heat resistant, as it turns out, not the vegetative part, but they have a tendency to be able to make something called endospores. So he proposed they were heat resistant, and Cone came along and said, oh yeah, actually there's endospores in some of these bacteria, and those are heat resistant components. All right, next, let's have a simple question, I hope. What are the 3 domains of life? Write that into the chat. That's right. Bacteria, archaea, and eukarya. Which of these domains contain microbes? That is right, all of them do. Bacteria and archaea are all microbial because they're all single-celled prokaryotes too small to be seen with the naked eye. And the domain eukarya, which does include us, obviously macroscopic, and fungi, which has some macroscopic mushrooms, and plants, which are all macroscopic, nonetheless has many members that are microscopic, too small to be seen. Some fungi are too small to be seen. And then there's a whole bunch of protists, both animal-like, like algae, and are animal-like protozoa, and plant-like algae. Are there any non-living microbes? If yes, list them. Obviously there are, otherwise I wouldn't ask. What are they? Right. Yeah. There are viruses. They're not considered alive, even though they can replicate and do have genetic material, but they don't have all of the qualities of a cellular life form. Viroids and prions, which are just infectious molecules and are not really considered alive. There's no cellular component. They're not complicated at all. They don't have any genes, but nonetheless, they can replicate themselves kind of like little bots. Okay, so here's an overview of the different categories of microbes that we'll consider. The living ones, bacteria, archaea, algae, protozoa, and some fungi, and as well as the parasitic worms in their larval stage, and viruses, viroids, and prions being the infectious non-living agents. All right, so we're just going to do a fun thing here. This is not something you have to know for a test at all, but it does help to remember that all the stuff we're talking about, even though it's hard to imagine sometimes, some of it's abstract. When we get to genetics and metabolism, you may struggle with that. Or maybe you won't, but it can be a little challenging because there's nothing to touch. It has to be mental. But everything we talk about is super relevant to things that you've already heard of before. So I am going to do a little cold call, and this is completely low stakes. Like, there is no reason for you to know the answer to this and to be right, and you don't have to be right. And we're not going to even know if you're right till the end, so you don't have to feel bad at all. But I'm going to cold call people, and you're just going to unmute and guess for the disease that I ask you about, whether it is bacterial, viral, a protozoan, or other, which encompasses, the other forms of microbes that might not be in this list. Things like fungus, things like prions, things like viroids, etc. Okay, so let's begin with ARIA and typhoid fever. What's your guess?
Bacteria.
Okay, great. Salmonella Anna, what do you think?
Bacteria.
Great. Mad cow, Brittany, what do you think?
Bacteria.
Great. Malaria Dane, what do you think?
Virus.
Great. Flu Emily, what do you think? Are you able to unmute?
Sorry, virus.
Okay, great. Gabby Rabies, what do you think?
A virus.
Great. Genna, polio.
Polio, bacteria.
Great. Grace, common cold, what do you think?
Virus.
Great. Gurpreet, AIDS, what's your guess?
Virus.
Wonderful. Jacob, syphilis, what would you guess?
Bacteria.
Okay, good. Jennifer, herpes, what would you say?
Virus.
Great. Jiho, gonorrhea, what's your thought?
Bacteria.
Excellent. Smallpox, Kimberly, what would you say?
Bacteria.
Good. Kiran pertussis, what's your guess?
Bacteria.
Okay, tetanus, Leslie J.
Bacteria.
Okay, wonderful. So let's see if we were right. And just so you know, like, again, just to be transparent about another reason I want to do this is I want you to remember that when people first discovered these diseases or figured out, oh, there is a disease called rabies, or I'm going to classify this as typhoid fever. frequently they had no clue what the causative agent was. And sometimes it was then subsequently figured out, and maybe it wasn't. Sometimes the person who originally sort of figured out what the disease was died before we figured out what was the causative agent. So, there's always a point of ignorance in learning. And you should never feel bad about that. Okay, so here are the answers. Okay, so many bacterial illnesses here, lots of viruses, but also protozoa that cause malaria. And then mad cow. Anyone know what mad cow is caused by? You can raise your hand if you know the answer. Emma, go ahead.
A prion.
That's right. An infectious protein. Crazy, right? All right, so next activity we're going to do is see how much you can remember about the different defining features of the different categories of microbes that you would have been introduced to in chapter 1. So here are the categories: bacteria, archaea, fungi, protozoa, algae, viruses, viroids, and prions. You want to think about how they are distinct from each other in terms of their size, composition, what are they made of, their structures, their chemical composition, and whether they're considered living or non-living. So just kind of make yourself a little grid of these guys and what features they have so that you be ready to answer if I was to cold call you, because that's what I'll be doing next. The reason bacteria is underlined is just to remind us that we are focused on bacteria in this class quite a bit more than the other microbes, but nonetheless, we're going to give everybody an equal shot this chapter. So just take a few minutes to do this and be ready to be cold called in case it is you that's on the hot seat. All right. So we're going to start our cold calling. As always, if you have absolutely no idea how to answer something, you can pass. But do let me know if you did write something down and what that was if I cold call you. And we're going to start with Marissa. We're going to go to bacteria, Marissa. What did you say?
I had it as like an average of 1 micrometer, which I think it says under the picture, if I could be wrong. And then also that they're single cell prokaryotic cells.
Excellent. And then we'll just ask anyone who has anything else to add to raise their hand to add to that excellent answer. I'm not able to see on your screen name. Is it Aislinn?
Yes. Also that bacteria are the only ones that contain peptidoglycan.
Thank you. And did I say your first name right?
Yes. Yep.
Aislinn. Okay, thank you so much. All right, excellent. That's great. And Aria, did you have anything to add? Can you? Yeah, go ahead.
Oh, sorry. I was trying to figure out how to unmute. It has 3 different shapes, usually bacillus, coccyx, or spiral shapes.
Great. Good job, you guys. All right, let's move on to fungi. For that one, I'm going to call on Mylin.
Hi, so for fungi, I have down it's larger, typically found like in mushrooms. And they are single. No, they don't have cells, right? They're single cell.
They are cellular, that's right.
Oh, yes.
And some of the larger ones, the macroscopic ones, are mushrooms. Good. All right, let's see if we have anyone who wants to add to that answer. Just raise your hand if you have something else that you'd like to mention. Laura, go ahead.
Sorry, they have is it chitin cell wall?
Yes, that's right. Their cell wall is made of a chemical called chitin. Fantastic. And the microscopic ones are not the mushrooms. The mushrooms you see in the forest are definitely within the kingdom fungi, but there are also microscopic ones that some of which look like mushrooms, but you wouldn't be able to see that unless you looked under a microscope. And others are like yeasts, which are single celled. So over here on the top right, you'll see some ones that look like mushrooms, but these are microscopic. This is a bread mold. You wouldn't be able to see it with your naked eye. Great. Okay, let's go to protozoa. And for this one, I'm going to ask Nicole.
Protozoas are single-celled, but they're usually bigger than prokaryotes, and then they also don't have a cell wall.
That's right. Okay, good. That's a good answer. Does anyone want to add anything to that answer? Raise your hand if you have anything that you put down that wasn't already said. Okay, so yeah, I'll just add a couple things. As mentioned, they are larger than prokaryotes, so that makes them eukaryotic. They are single-celled. They're animal-like in the sense that they move around and they do not have cell walls. And so you can see that here. This is an amoeba. That's an example of a protozoan. All right, for algae, we're going to go to Oscar.
They can be single or multi-celled. They are photosynthetic. They usually live in the water. Their cell wall has cellulose, and they may have a flagella to move around.
Excellent. Yeah, all good answers. Fabulous. If anyone has anything they wrote down that was not already stated, go ahead. I'll look for your raised hand if you have anything you want to add to that. All right. Yes, great job. Next, we're going to go to viruses, and let's ask Pia, what did you write down about them?
Can you hear me?
I can hear you.
I said that they're acellular.
Right.
And they infect the host cell. All forms of life are affected. And their structure is they have the core is either DNA or RNA. It's never both of them.
Right.
And then
yes.
They also multiply using host resources.
Yes, good job. All right. Anything anyone wants to add to that? I'll just pause for a quick raise your hand. All right, good job. All right, viroids are next. And for that, we're going to ask Ronette. What did you write down about viroids?
I said that they're simpler than viruses. They require the host to replicate, and they cause plant diseases.
That is right. Excellent. Anyone want to add anything at all to that? Just raise your hand real quick. Emma, go ahead.
They only use RNA, they don't use DNA.
That's right. They're made of RNA. That's right. Good job. Okay. Last but not least, prions. And for this one, I'm going to ask Sunday.
I didn't get to this one, but I think so. It's only proteins, no DNA or RNA.
That's correct.
And then like when they go bad, it's from the like abnormal shape or whatever they fold.
Yes, good job. Anyone have something that they want to add to that? Excellent answer. Raise your hand if there's anything you want to add that wasn't already stated about prions. Okay, I'll just add something real quick. They only, as far as we know, infect animals, and that includes humans because we are animals, in animals that have a brain or a nervous system because they get into the neurological tissue. All right, so here is kind of a cheat slide, if you will, to help you study kind of what are the different sizes, what are some of the key features, what is, distinctive things about them might be what their cell wall is made of, or that they don't have a cell wall, what cell type are they, prokaryotic, eukaryotic, or acellular, what composition do they have, etc. And Laura, your hand was up. Did you have a question or comment?
Yes. Will you make these slides available to us?
Yes. So did you see the pre-post yet?
From the Zoom?
Yes.
No.
Okay, great. I will review that at the end of the class, but just sort of, I'll say it in words now. If you go into Canvas in the weekly module in the topic section, okay. Right next to the page where you do your prep for the Zoom, after that is a page with a date. It says Zoom, today it'll say Zoom Wednesday, January 5th. And if you click that the night before class, there'll be a slide deck that is not complete, but has a skeleton of what we'll talk about. And then I do post these completed slide decks about a week before each exam.
Okay, awesome. Thank you.
Yeah, great question. All right, so let me have a look at our time. I'm also going to look at where we are in the slide deck to see if I'm going to be able to do this. I think we do have time for this. Okay, so what category of microbe is COVID-19 belong to? Write that into the chat. Right, that's correct. So the disease COVID-19 is caused by SARS-CoV-2, which is the virus. And what features of COVID-19 make it part of the virus group? I'd like you to take a second to maybe use your phone or your computer to look up what is known about the structure or the features of COVID-19. And then, so you have something to sort of say about this, what features make it part of this group? Thinking about what we said, what makes a virus, and what about COVID-19 makes it a virus? So just take a second to do some research or search your head if you already know lots of things about it. And then I'll prompt you to answer in a second. Okay, so what we're going to do right now is just I'll raise your hand to just say something about that you know or found out about SARS-CoV-2 that is sort of makes it part of its group, one of the features or things that you know about it that make it part of this group. So just raise your hand if there's anything about it that you know makes it part of this virus group. Jennifer, go ahead.
It's a virus, so it infects the hosts.
Yeah.
And it replicates and mutates as it, keeps getting passed on.
Yes. So it's non-cellular, but it infects host cells. That's a definition of a virus. And it does have genetic material, so it does mutate during its replication. Good. Anyone else? Something about COVID-19? You can think about all the things that make a virus, like its size, its chemical composition, it's what we just talked about, very important. It's like replication strategy. Anything else that comes to mind? Heather, go ahead.
Is it just RNA?
It is made of RNA, although it also has a protein coat around it. But yes, its genetic material is just RNA, as opposed to our cells and other cells in general. Cells have both RNA and DNA, but viruses will only have one of the nucleic acids, and COVID-19 or SARS-CoV-2 only has RNA as its genetic material. So yes, that's a good answer. Aislinn, what would you like to add?
Something that seems to be specific to COVID-19 is that there's sugar molecules actually coating the spike proteins, which makes it more difficult for the immune system to recognize them.
Yeah, so it has particular molecular signatures on its surface that have specific interactions or specific consequences in the human body. Yeah, excellent. Mylin, what would you like to add?
Isn't it part of the family of SARS as well, and that's considered a virus?
Yes. So if we analyze its genetic material, if we read the letters, we see that it is very similar to other viruses that belong to the SARS family. And so we can say, yes, it's a virus, and here's its family that it belongs to. Good job. Okay. Good work, you guys. Okay, so it is a virus. And one of the things that was mentioned was the composition being RNA as its genetic material. Viruses, and COVID-19 is no exception, also have a protein coat around them. It's considered to be non-living, as was mentioned, so it's acellular meaning not a cell. but it is able to replicate. What about size? Did anyone find out what its size was? Or know anything about what its size is? Maybe you can look that up real quick. And if you find out, you can raise your hand. Vict, go ahead.
From my research, it says 0.1 micrometers in diameter.
Yeah, 0.1 micrometers, which is to say 100 nanometers. And that is a viral size for sure. And I love that, Jennifer. Yes, teeny tiny. Correct. Okay, so you have a little bit of time before the break. And what I'd like to do with that time is do what I like to call the what's this game. I'm going to give you some information. You're going to try to decide what something is. Okay, so first here is the scenario, okay, the pretend scenario. So this member of the military, he's stationed in West Africa, he develops a skin ulcer and the army doctor looks at it and he says, based on his experience, it's maybe this cutaneous leishmaniasis or this cutaneous anthrax. Cutaneous is a word meaning skin. So it's either anthrax or leishmaniasis, both of which are microbial diseases. So here is the private's wound. And here are some pictures of people with both of these conditions. So here's his skin ulcer, and this is leishmaniasis in other people. It's caused by a species in the protozoan group in the genus Leishmania, and it's carried by sandflies when they bite you. On the other hand, this is cutaneous anthrax caused by a bacterium called Bacillus anthracis. This is something you can get by handling goat skins as one possibility. Okay, so I am going to ask you in the chat, I'd like you to make a guess as to which one you think it is. Great. So we have some people rooting for team leishmaniasis and some people rooting for team anthracis. If you were on the leishmaniasis team, I'd like you to raise your hand just so we can find out what your reasoning is. And if it was a total guess, that's also fine to use as a reasoning. So we're clinical doctors. We don't know the answer. We're trying to figure it out. If you had two doctors in the room and this was the two possibilities, one might say one might say the other. So we've got to start from somewhere. So let's start with Mylin. What is the reason that you are on team leishmaniasis?
Can you hear me?
I can hear you.
I picked it just by a wild guess because he's in military. And so maybe being overseas, we don't have sandfly bites.
Okay, so yeah, so your reasoning is maybe it's you're taking the cues from what his likely exposure is. Okay, good. All right, Heather, you were also on team leishmaniasis. What was your reason?
Pretty similar. I was trying to think about, where he's at and kind of like what he's going to handle. I didn't really see him handling goat skins, so.
Okay, great. And Aislinn, what's your reason for being on team leishmaniasis?
Also more or less the same because being out in the field, especially in the military, more likely to be exposed to insect bites, other creatures not necessarily domesticated.
Great. And then I think we have Jennifer. Did you also have a reason? I wasn't sure if that was left from before, but if you're on team leishmaniasis, tell us why.
I'm not, sorry.
Okay, great. All right, so now let's have team anthrax weigh in. If you have, we're just going to talk to like a handful of people, but if you're on team anthrax and you want to share your reasoning, let's go to Sunday first.
So I based mine off kind of like the color of the wound, but I can kind of see why people were saying the first one, because like the sandflies, and he's most likely to be infected, I think, by that one now.
Great, but your original thought was, oh, it looks more similar.
Yeah. Mm-hmm.
So you're basing it on like sort of the visual inspection. Okay, great. Sophia, why don't you tell us what your reason is for being on team anthrax?
I'm actually
with Sunday, I also compared the colors because if you look at how the first one looks, it's more red than dark. So I just compared the colors, how anthrax looks darker in the wounds than from the sandfly bites. So I just figured that maybe it's the anthrax. I never really thought about like the place and where those flies could be.
Great.
Because
Fabulous.
maybe like once he like did handle a goat and he got it. I don't know.
Yeah. Okay, wonderful. So this is perfectly illustrating the exact, conversations that might be happening in the clinic. Now, if we really want to know the answer, then we've got to start asking questions that can be answered with a test. Okay, so we've got to sort of say, okay, well, what kind of things do we need to know to really nail this down? So if we want to diagnose him, we have to know some things about these two different causative agents. So leishmaniasis is caused by a protozoan, in the genus Leishmania. Anthrax is caused by a bacterium, Bacillus anthracis. Okay, so based on knowing this, additionally, what information about the size, chemical composition, metabolism, or internal structures of the organism that's actually infecting this guy, okay, would help you to know which one we're dealing with? So you can put a question into the chat. You can ask me anything that is testable. You could ask me, what's the size of this organism? You could ask me what type of metabolism does it do? You could ask me, does it have internal structure X, Y, or Z? You can ask me things about its, does it have a cell wall? What is the cell wall made of? So ask me a question in the chat and I will answer it. Okay, so we have, it's kind of like a 20 questions here. Laura says, does the cell have a cell wall? Okay, the answer is yes. And Emma asked that as well. Mylin says, do you have more than one lesion? The answer is yes. What other questions do you guys want to know? What is the cell wall made of, Gabby says? Oh, that's a good question. The cell wall is made of peptidoglycan. If at any point the answers to these questions make you think that you know now the answer for sure and can defend it, go ahead and raise your hand. All right, Emma, go ahead.
I think it's bacteria because that's the only one that has peptidoglycan in the cell wall.
That's right. So based on those simple questions that, something we could probably simply test in a lab, we can narrow it down very quickly. We can say, oh, it has a cell wall. Well, protozoa don't have cell walls. And we know that bacteria have peptidoglycan in their cell wall. So I want you to start thinking about what kinds of information this class is going to provide that will enable you to get past the initial questioning that anyone would be able to say, well, I am paying attention to his intake information, where he was exposed. That's important. And I'm paying attention to the visual look of this. That's totally important too. But being in this class will allow us to ask us more sophisticated questions that really put the nail in the coffin of a diagnosis. Okay, so we have a diagnosis. We think it's Bacillus anthracis. We think he's got anthrax. Maybe he handled some cured goat skins, which happen to be around in Africa, all the other places as well. It could have been the other one too. Okay, just that I wasn't like trying to trick you. It could have been the other one too, but in this case, it was Bacillus anthracis. All right, let's do another one, and then we'll take our break. So Maria, she's a resident locally at the Washington Oaks assisted living facility. She gets pneumonia. We need to diagnose her so that we can give her the right treatment to keep her alive. What type of microbes can cause pneumonia? Put that into the chat. What type of microbes can cause pneumonia? Bacteria, viruses, yes. Mycoplasma. Fungi, yes, those are all possible. So a lot of different microbes. So viruses, fungi, and bacteria are the primary causative agents of pneumonia. And those are the ones we're going to consider in this example. Okay, so now I want you to ask a question in the chat, just like before, that will help you to determine what we're dealing with. And I'll answer it. And as soon as you guys think you have an answer, you should raise your hand and be able to defend it. So ask me a question in the chat and I'll answer it. Okay. Does it have a cell wall? It does. What is its cell wall structure? The cell wall is made of chitin. What's the size of the microbe? It's about 10 micrometers long. Is it acellular? It is cellular. Emma, what's your answer?
I think it's fungi because the cell wall is made of chitin.
That's right. Okay, so it was very quick, wasn't it, to get to this diagnosis? Now, in the real world, some of these questions might take longer to answer than 2 seconds, but hopefully you start to get the idea of what I'm getting at here. What I want you to focus on is the things that you'll learn in this class that will be helpful in this regard that you might not be thinking about, you know, or which you can now add to a conversation with a person who has not had microbiology as their background. Okay, so we are going to take our 10-minute break. Right now it is 10:54 or 10:55. So let's come back at 11:05 and we're going to do another little activity together. All right, I'll see you at 11:05. What's up?
Is it possible if I could talk to you really quick in a breakout room? Or would you prefer after class?
Do you have time after class?
Yeah, I can.
That's perfect for me.
Thank you.
Yeah, no problem. All right, welcome back, guys. I need to stop screen sharing because my screen is doing something really wackadoodle right now. So I need to fix that. Hold on 1 second. Okay. What is going on with my PowerPoint? It's just gone into some very strange mode. There we go. Much better. Phew. Okay. Okie dokie. So we're next going to tackle this question, which we did discuss in the first day, which is why do microbes matter to you? So I'd just like you to write a reason into the chat. Why do microbes matter to you? In this context, unlike on a test or in a lab, then you could write anything, including I have to pass this class to move on to the next part of my life. But do try to think about it from the perspective of what this class is teaching. Okay, so microbes make food, that's right. If we want to treat disease, microbial disease, it's important to know about microbes. They are everywhere. Yep. Some of them can help us with new technology or to develop medicine. If we are trying to identify diseases, it's important to know about them. So, you know, without knowing what microbes are, it'd be hard to identify which one we're dealing with. They live on us. They're part of our ecosystem. Yes. Yes, they contribute to a healthy gut. And yes, they are capable of lots of different forms of metabolism. So they manipulate our environment. We can use them to manipulate things, including, yeah, gas fixation. And excellent. Okay, all great answers. So an important thing that I hope you take away from this class is that not all microbes cause disease. And in bacteria in particular, not all bacteria are disease-causing agents. Actually, most bacteria and most microbes are not pathogenic. However, the ones that do certainly have a disproportionate impact on our lives. And more people have died of microbial disease than have died in any war in human history. We have normal microbes that live on us and on our mucous membranes and on our skin. And that's part of our normal microbiome. That's actually also part of our immune system. We have microbes that are important for our ecosystem because their metabolic activities are ones that only they can perform. So decomposition and fixation. So there's sort of opposite processes. Decomp, taking stuff and breaking it down so it can be reused. And fixation is taking stuff that's inorganic and turning it into organic molecules. Some are domesticated. So microbes help in food production. Some of them help in industrial processes. And some of them have been manipulated to produce useful things for us or to help us learn important things. So we do not know everything yet. And part of the reason for that, as it turns out, is that fewer than 1% of all microbial species can actually be cultivated. There are ways to study microbes without cultivating them, but it is more challenging for sure. When we developed sequencing technology for bacteria in particular, that's when we really learned how much we don't know. Because sequencing allows you to know what's out there that has genetic material, at least, without having to grow it in the lab and study it in culture. So just as one example, a small sample of the Sargasso Sea, which is also known as the Bermuda Triangle, revealed 1,800 new bacterial species within that sample of water that had never been seen before. And any one of those species has potential new uses in industry, medicine, science. So there's a great deal of scope for new discoveries still in this field. At the same time, the things about microbes that are challenging or relevant remain to be understood. So diseases are arising all the time that are microbial in origin. Those are new challenges. And the environment is heavily dependent on microbes. Sort of understanding their exact role is not fully explored. So we have lots to learn still. Some sort of highlights of the past, I guess. Studying microbes has allowed us to eradicate plagues. For example, a worldwide effort to provide a very effective vaccine against smallpox resulted in the eradication of this disease from the planet for the first time in human history. And it's probably a disease that's been with us certainly for all of recorded history, and there's a lot of evidence that it was with us before then. And another plague that you've probably heard of, the Black Death, has been well controlled with antibiotics, though it isn't gone. But because we now know how to deal with it, it's pretty much, as long as you have access to antibiotics, no longer a threat to human life. So with that said, there's lots of challenges remaining on the disease front. So things that used to be common to infancy, a lot of people died in infancy of respiratory infections or diarrhea, still happens worldwide. It just depends on where you live and what you have access to. And there's a lot of people impacted by disease in all parts of the world, as we have all been dealing with the pandemic for several years. We certainly are intimately familiar with that. And there's new diseases arising all the time. Some of them are brand new. They've crossed into the human species from animals for the first time. Others are not brand new, but we had them under control and then they change, and that brings them back into focus. Okay, so we're going to take a minute now, or more than a minute, to do a Kahoot. I don't have it loaded up yet, so that gives you a chance to go and either open your app or go to kahoot.it and get logged in. And I'm going to take a moment to get my Kahoot loaded up and running so you can get prepped while I'm doing that. Okay, so I'm going to display the code. Again, this is the code that you're going to put on into the Kahoot to join this game. It is 660-7005. So please log in and let's get started. All right. So as always, this is the game pin to join. And if you get logged out, you will see it displayed at the bottom. You can always rejoin the game. But let's get started. What best describes the contribution of this group of scientists, Tyndall and Cohn? That's right. They hypothesized and/or discovered endospores. What best describes the contribution of this group of scientists, Jenner, Ehrlich, and Fleming? That's right. They were all involved in developing antimicrobial interventions. Which group investigated germ theory, the theory that specific microbes cause disease? Bossi, Semmelweis, Pasteur, and Koch. Good job. Which domain contains microbial members? Correct. It was all of them. A microbe 100 nanometers wide made of RNA and protein is probably viral. That's right. A microbe with a cell wall composed of chitin is probably fungal. Yes, indeed. 0.45 millimeters is equal to what? Yes, 450 micrometers. 0.45 millimeters, millimeters being larger than micrometers. So to get to micrometers, you have to multiply by 1,000. True or false, most microbes cause disease. That is false. Most microbes do not cause disease. Most microbes are not pathogens. All right, let's see who made it onto the podium today. If you didn't make it onto the podium, I know that you were right behind the front runners, so you should feel proud of yourself and thank you for participating today. Okay, good job, everybody. All right. Back to our slides. These Kahoots will be posted for you to practice about a week before the major exam. You'll see a page called Unit 1 exam tools or something like that, that has the Kahoot links on them that you can use as practice. All right, so what I want to spend the last half an hour talking about is lab. There's several things I want to cover. First, we will be doing a project this quarter called the Unknown ID Project. This is standard in a microbiology course. You're given a microbe, a bacterium, a culture, and you have to figure out what its species is using a variety of tests. Because of the restrictions due to the pandemic, we are going to do this in a digital format. So the only thing you have to do this week is sign up for an unknown ID number, and I'll show you how to do that in a second. This very first lab also involves some calculations. So you're going to be calculating resolving power. And I'm sorry, let me restate that correctly. The first lab that you're doing this week, which is the online only lab, involves calculating CFUs per square foot hour and CFUs per mil. And I just want to briefly review that with you. And then lab 2, which we're doing next week, involves calculating resolving power. And so I want to preview that with you because you'll need it for the pre-lab, which is due on Sunday. All right, so the Unknown ID project is going to take you several months. And the very first step is to sign up for a number. That number is assigned to a specific species of microbe, but you don't get to know what that is. The technician knows. I don't, and you don't. So what you're going to do is do a series of steps over the quarter to figure out what you have. And the first step, which will take place next week, is you'll figure out its gram status and its morphology, a word that means shape. That will then lead you to a dichotomous key that will allow you to do part 2. And through a series of between 2 and 5 metabolic tests, you'll be able to identify the group to which your microbe belongs. After that, you'll do a series of further testing, much more extensive, probably 10 or 20 tests that allow you to figure out its exact species. And then you write a lab report. So I do want to show you on Canvas where to go for the Unknown ID sign up. So let me just share my Canvas course shell with you. In the modules, which is where you should always go, there is a couple places that you'll see it. So in week 1, it's mentioned in the overview, and it's also mentioned in the lab, prepare for the Unknown ID project. And in the due this week for this weekly lab, you'll see an Unknown ID sample sign up. What it is it's a wiki page, and you're going to add your name to one of these numbers. Okay, so some people have already started doing that. You should add your numbers as well. All right. Next, where did I put it? I want to show you one other thing. So if you're curious about the whole project, and you should start reading as soon as you can to kind of prepare yourself for it, there is a module which has all of the information about the Unknown ID project in it, and that's down here at the bottom. So you can just, here's the wiki page, but you can start reading about background on this project and the resources page and so on. All right, to the slides. And so the deadline for the sign up is Sunday. Last class, we talked about how to calculate quantities of colony forming units, so cells that land on an agar plate and then grow into a colony, in air and water samples. And so I just have a couple slides to review with you so that everyone is really on the same page. So I'm just going to review this one and remind you how to use this as a template to practice. So here's a plate. And because it's got so many, it's actually, it's not just when you count this plate, you're not going to come up with the same number as everybody else all the time. And you're welcome to do a form of estimation when the plate is very dense like this. What I like to do is divide the plate into sections, count one of the sections and multiply back. So let's suppose I was like feeling especially tired and I was like, I'm only going to count a quarter of this plate. So I divide it in fourths and I count a quarter of it and multiply by 4 to get my estimate. And so your estimate's going to end up being around this number, but it could be different. And every time you do it, it'll be a little different. So that's good. So, have whatever that number is that you calculate that moment. You can adjust what the surface area of this hypothetical plate is. You can decide it's going to be a 1/5 or a 1/2 or a 1/3 or a 15th or a 20th, depending on, just that moment. But in my little practice, I'm going to say, suppose this plate was 1/8 of a square foot. You would then need 8 of these plates to sample a whole square foot, which is what you want. But a simpler way to do that is to count the one plate and multiply by 8. You can expose in your example, you can expose for any amount of time. I'm going to say it's 10 minutes. What fraction of an hour is 10 minutes? Put that into the chat. Correct. It is 1/6 of an hour. Therefore, what should we multiply this number by in order to get what would have fallen on this plate in a whole hour? Correct. You could multiply it by 6. So we can take this number, we can multiply it by 8 to account for the area and by 6 to account for the time. And if we did that, we would get a number, a CFU per square foot hour that's 4,800. But by adjusting these numbers, you can say, well, what if it was a 20th of a square foot? And what if the exposure time was only 5 minutes? Then how would I do this math and run through it? Any questions about this right this second? Okay, quick reminder about the CFUs per mil, how to use this slide. Again, you'll see that there's sort of a, how do we count the exact number? Like, is this 2 or is this 1? And how many are in this line? So you're just going to do your best, okay? And, it's going to be in the realm of 30, but it might be bigger or smaller. You're just going to count and get a number. And then you can adjust these volumes and you can adjust these dilutions. So whatever this is going to tell you what to do, but you can put new numbers in and solve it in practice. So in my example, I'm going to say, well, what if the volume used was 0.3 mils? And what if the dilution was 1 in 10,000? Then I would take my CFU number and I would divide by the product of the volume times the dilution, which gives me 1, 2, 3, 4, 5, a million CFUs per mil. I had to actually do the math and count the numbers. The other way you can do this is you can figure out what fraction of a millimeter this is and multiply by a number to account for that fraction. And you can figure out, you can just multiply by 10,000 here to get your same number. Okay, so to test whether we understood this, as well as I hope you will eventually understand it, I'm going to again go back to the Kahoot page, but this time we're going to go to a CFU practice Kahoot. Just see. And again, don't stress about this too much. You're not being graded on this, so just do your best. It's going to go by kind of quick, but it's a way to kind of practice when you're under a time pressure, which is good. All right, so let's sign in for this Kahoot. The pin is coming up here in a second. There it is, 805-4044. 805-4044. Great. All right, let's get this party started. And again, if you get kicked out of the Kahoot and need to rejoin the game pin will be displayed at the bottom. If you can't get into the Kahoot at all, you're always welcome to put your answers into the chat. All right. To quantify microbes in an air sample, what do we calculate? Here's your game pin at the bottom if you need it. To calculate microbes in an air sample, we calculate what? CFU per square foot hour is correct. That's right. Quantify microbes in a water sample, we calculate what? CFUs per mil, good job. How many CFUs are on this plate approximately? It is 25 is the best closest number. A 15 centimeter plate that is 1/5 of a square foot, also 0.2 of a square foot, was exposed to indoor air for 30 minutes. What is the CFU per square foot hour? This is the same plate as before, just so you know. Good job. 250 CFUs per square foot hour. How did we calculate that? Well, the plate had 25 colonies on it. It was 1/5 of a square foot, so you multiply by 5, and 1/2 an hour, so you multiply by 2. Or you could have taken 25 divided by 0.2 times 0.5. Either way, you would get 250. How many CFUs are on this plate? $17. All right, so if this plate was spread with 0.1 mils, also known as 1/10 of a mil, of a 1 in 1,000 dilution, what was the CFU per mil of the original sample? $170,000 CFUs per mil. So we take our 17, we say we only spread 1/10 of a mil, so we've got to multiply by 10. That gives us 170. And we diluted 1,000-fold, so we should multiply by 1,000. That gives us 170,000. The other way you can do this is 17 divided by 0.1 times 0.001. And either way, you're going to get this number. All right, good job, everyone. All right, so let's go back to our slides. Talk about the last little bits of things that I want to talk about today. All right, so one of the things that you're going to be looking at in lab 2 is microscopy, and one of the major concepts is resolution. Resolution is a term that can be defined lots of ways. You may have heard in a previous class that it's called, one of the definitions is the smallest distance between 2 points that can still be seen. In this class, the most useful definition I have found is the smallest thing that can be seen with a lens. So resolution is a way to define what you can or can't see with a particular microscope or the lens of your eye or an electron microscope. And there is a way to quantitate that, and it's called resolving power. And resolving power is accomplished by an equation. If you have a light microscope, you can use that equation to figure out the resolving power, which means basically the smallest thing that can be seen. So our human eye has a limit of resolution or resolving power. That means that we can see down to things that are 200 micrometers or 0.2 millimeters. Of course, this is an average. Some people with very exceptional eyes might be able to see slightly smaller things. That's unusual. Most of us, as we get older, can no longer see this close. I certainly can't. My number would probably be horrible. Light microscopes, typically a good light microscope is about 1,000 times better, which means it sees things that are 1,000 times smaller, 200 nanometers, 0.2 micrometers. And an electron microscope is even better, more than 1,000-fold better. So it can see things that are less than 200 picometers. So this resolving power number, is it better for it to be a small number or a large number? Put that into the chat. And if you're not sure, put a question mark. That's absolutely fine. Let's wait for everyone to weigh in. I really want you to think about this. And even if you don't know the answer, you just put a question mark. I assure you putting a question mark is better than not participating. It's going to train your brain to pay attention to this thing. If you don't know, telling yourself you don't know, it's going to make you want to remember that information. Okay, so there's a bunch of question marks. Some people have chosen smaller. I would say the majority of people chose larger. That would be your gut instinct. That's the gut instinct of most people. In this case, your gut instinct is dead wrong. Okay, there's no shame in that, but it's not correct. So in this particular instance, remember that resolution is the smallest thing that can be seen. We want to see the smallest thing possible. So a resolving power that's a really small number, like 200 picometers is teeny tiny versus 200 micrometers, is way better. Does that make sense? Yes, no, or kinda? Great. Okay, so for now, if you're not completely sold on the idea that a smaller number is a better resolution, then just memorize it. And then hopefully by the end of the week, you'll be convinced. Okay, so let's talk about how to calculate resolving power. So there's an equation that tells us how small something can be and still be seen by a particular microscope, for example. And that equation is given in the Lab 2 materials, and it's called resolving power, or RP, is equal to the wavelength of incident light divided by 2 times something called the numerical aperture. So you can memorize this equation. It's very simple. There's only 2 things you need to know: the wavelength of light and the numerical aperture, both of which would be given to you in a question. But what is the wavelength of light? Well, visible light is on the EM spectrum. And so we can't see things like gamma rays, X-rays, and UV light because they have a much shorter wavelength than our eyes can detect. But around the violet blue spectrum, we can start to see light. Wavelengths get larger and larger until we hit the red side. And then after that is infrared, also heat, and then radar and radio. So the visible spectrum of light, the wavelength is really small comparatively on the blue side and really long comparatively on the red side. The wavelength itself helps explain whether or not you can see something or what it affects resolution. So looking at this equation, knowing that we want our RP to be small, a small number, okay? We want our RP to be a small number. And looking at how this fraction is set up, we want this to be small. Wavelength is on top. Do we want our wavelength to be small or to be large? And if you're not sure, putting a question mark is a totally legit answer to this question. But if you do have an idea, then put your idea in. Great. So because we want resolving power to be small, we want our wavelength to also be as small as possible. Which wavelength is going to be more helpful, blue or red, if we want a small wavelength? That's right, blue, okay? So the closer we are to the blue side of the spectrum, the better. And that will be easy to tell because I would give you the wavelength and the number would tell you. So, you know, the blue side is around 400 nanometers and the red side is around 700. So the smaller, the better. Why is that? Well, just very sort of simply to kind of cut through a lot of physics and be slightly inaccurate, but hopefully enough of an answer. If we look at the wavelength of light, it kind of tells us how much of the sample is being hit by the light. And so if we have a really short wavelength, then a lot of different points in the sample are being hit. So they're each giving you information. As opposed to really long wavelengths, it's only being hit in a couple different places. So you don't get as much information about the sample. It's the best way to think about it. So to get a smaller RP or better resolution, we want our wavelength to be as small as possible. Now, NA is a unitless empirically derived number. It's just given on a lens. It's basically, you create this glass lens and then you test it, and that tells you how good it is at resolving things. And so then when you test it and you figure out, like, you know what this is, actually, it's kind of back engineered. You know what this is, so you figure out what this has to be to make that the right thing. And then you put that number on the lens. So I would give you that number. It doesn't have any units. You don't have to like memorize it or anything. I would just tell you. But we can see different lenses, typical different lenses have these for a variety of NA values. So if we look at this equation again, right, we wanted our wavelength to be small so that our RP could be small. Do we want our NA to be small or to be big? Right. Because it's in the denominator of the fraction, we want our NA to be as big as possible. That's going to make this smaller. So we want a large NA. Okay. So if these were four lenses that you had the option to decide between, which one would you pick to get the best resolution? Right. You'd pick this last one, the 100x, because it has a large NA. Now, what if there was like a 200x? Okay. This gives you better magnification, but its NA was 1. Would that one be better or worse than the 100x? Right, it would be worse because even though the magnification is better, the resolution is actually worse. Okay. So we're going to do a quick example. Let's see what my time looks like. I have 5 minutes. Let's suppose we had a 15x ocular and a selection of objective lenses, an 80x oil with an NA of 1.2 and a 100x air with an NA of 0.5. Okay, so this one has better magnification than this one, but it has a lower NA than that one. And then we have 2 light sources to choose between. We could use yellow light, which has a 600 nanometer wavelength, or green light, which is a 500 nanometer wavelength. So I'm going to dispense with the total magnification question for now, because we haven't really talked about that, and you'll get a chance to look at that in lab. But which combination of objective and light source would give you the best resolving power? So you have to pick one of these lenses and one of these light sources. Which one is going to give you the best RP, allow you to see the smallest things? Right. So you want your wavelength of light to be small. So that's going to be the green light because it's smaller. 500 nanometers is smaller than 600 nanometers. And you want your NA to be as big as possible. So even though this 80x oil doesn't have as good of a magnification ability, it does have a better resolving power. So you're going to pick this one. I'd like you to do the calculation really quick and tell me in the chat what the resolving power is with this combination. Remember that it's wavelength divided by 2 times NA. Right into the chat if you do that calculation really quick, what the resolving power is with that lens and that light source. Right, good. So what we would do is take 500 and we would divide it by 2.12, which is 2.4. So 500 divided by 2.4, and that gives you 208 nanometers. And what does that mean? What that means is with this lens and with this lens and weight and light combination, you can see things that are 208 nanometers or larger. Okay, so what do we have to do next? By tonight, the orientation module is due. Orientation quiz, lab safety quiz, and your lab preferences survey if you want me to take your preferences into account. No guarantees, I'll do my best. By Sunday, there's a review quiz, unlimited attempts until the deadline, but after that, the answer key post, so no makeups for any reason, okay? Even if your house gets taken by a twister. Lab 1 deliverables, so make sure you fill out the Lab 1 wiki, put your stuff into the Dropbox that's asked for, and finish this post Lab 1 quiz. And then make sure that you read the Lab 2 background and instructions pages so that you're able to submit pre-Lab 2 by the end of day Sunday. And finally, sign up for a number on the Unknown ID wiki. Just a note, as looking at the course shell just now, I noticed that I had made a little bit of a boo-boo on my wiki. So I might be reassigning a few numbers, but I will let you know if that happens to be the case. And then before Monday, you make sure that you have done the prep for Monday. We're going to talk about microscopy. So there's a pre-quiz to do, there's a chapter to read, and most important, there's some video lectures that will take you several hours. So make the time and some slides to look at. Okay, so with that, I'm going to end the recording and take your questions. And I know that I have at least 1 or 2 students who need to talk to me privately. So I will take the general questions first. And if you want to talk to me one-on-one, stick around for after that.